Very good. Good morning, everybody, and welcome to today's lecture on the single-cell omics network in Germany. Before I introduce the speaker today, I just need to make a couple of announcements while waiting that everybody's sitting in front of the cup of coffee or breakfast at home or whatever that is that you guys have around. So this series of lectures, as I said, the single-cell omics network, which I'm also part of, is organizing. So it's really nice that you guys are popping up this morning. And I have to make just a couple of announcements. As I said, the next lecture will be in 4 weeks' time by Boyan, so please stay tuned. And the other two announcements are upcoming workshops. The next one is going to be held on November 17th, and this is hosted by John Walter, and this is on single-cell epigenomics. And the second one, which is an initiative by Lifetime and the VIB, this is an emerging technologies in single-cell research. I think you're seeing that right now on your screen, and this is November 19th. So please stay tuned. You will find all this in the internet. I'm going to start now introducing the speaker today, and it's really a nice pleasure to introduce Bonev to you today. I have seen Boyan's work over the last years, and I'm really happy to introduce him to you today. So Boyan did his PhD with Nancy Papadopoulou in Manchester, after which he moved for a 1.5-year postdoc with Paola Arlotta and John Ring in Harvard. And then after that, he decided to join Giacomo Cavalli's lab at the CNRS in Montpellier in France, where he really developed quite a number of very important findings, I would say, in our understanding of how the genome falls in 3D and how that is important for cell fate identity, kind of, let's see. And I'm sure that Boyan will develop much more on that. I will just mention that he's been in the Pioneer campus at Helmholtz Munich for a bit more than 2 years now. And I think with that, I have just 2 more logistic things to say. One is to remind you that the lecture will be recorded, and this will be posted on the website if you want to look at it later. And the second one is to ask you to please raise your hand at the chat, so at your controller, if you have any questions. And then after Boyan finishes his lecture, then I will unmute you guys to based on your raising hands. So with that, I think that's everything that I have to say. Thank you, Boyan, for being with us this morning. I very much look forward to your talk.
Good morning, everyone. Thank you, Maria Elena, for this very nice introduction. And thank you, everybody, for tuning up this early in the morning. I'm really excited to talk to you about today, about some of our latest work in the lab, which combines some of the aspects that Maria Elena mentioned before. So looking at the 3D genome and the high order interactions within the nucleus with exciting single cell technologies that are one of the major focuses in this network. And I'm going to try to convince you today that the integration of these two types of analysis can actually tell us a lot more in terms of how cell fate decisions are made in the cell. So I'm going to start by something that I'm sure most of you are familiar, which is the Waddington landscape. And in the last decade, especially with the advent of single cell technologies, there's been a lot of increase in our knowledge of how different cell types, how different cell states populate this kind of landscape. And we've learned to quantify and precisely determine how many cell types are there, in what particular interactions between them exist, and so on. But some of the aspects that I find fascinating, and they're still not very clear in this kind of landscape, is what is actually the nuts and the bolts, so the major building blocks that determine what particular path a certain cell will take. So the gene regulatory networks that underlie cell fate decisions during development are, I think, very fascinating and are one of the major aspects of the research in my lab. So I just wanted to give you a brief introduction of the different types of molecular layers that we think that the majority of these gene regulatory networks converge into. So I'm sure that most of you are familiar with the two major types of chromatin, so the heterochromatin, the eochromatin, but also as part of the epigenetic landscape, especially in mammals, DNA modifications such as DNA methylation play a very important role, and how all of these elements on the linear genome are actually close together in the 3D proximity is also recognized to be very, very important. So in the last couple of years, looking at the accessibility of chromatin, especially for regulatory factors such as transcription factors, has become a very important way to measure the epigenetic landscape. But what I'm going to argue today is that it's not the only thing, and looking at the different types and other types of molecular layers gives you an information that may not be contained just in accessibility. However, I'm going to start with a slightly bit more introduction on the 3D genome architecture, because I feel that this is important for our understanding of how exactly gene regulation occurs. So for those of you who are not in the field, especially in the last 8 years or so, there's been a huge increase of our understanding of how the genome folds. So I'm going to talk today only about two aspects of the genome architecture, so two different levels of architecture. So one of them is the topologically associated domains, or also called TAT. So these are structures that were identified about 8 years ago. And what they represent is essentially, you can see these triangles in the left picture, where there is an increase of interaction of regions that lie within those, and reduction of interactions that cross the TAT boundaries. And we now understand a bit more how these structures are formed in the genome, and these are mostly by the presence of CTCF and cohesin, which lie at TAT boundaries and seem to at least inform how the genome will fold at this level. However, what is also very important is that there is an additional within for example, one such TAT, we can find many different types of chromatin loops that exist simultaneously. So you can have an architectural loop, for example, also mediated by CTCF, like you can see here, but you can also find different types of regulatory interactions that may or may not be dynamic, and such as enhancer-promoter interactions. And whether or not these interactions are static and exist at the same time, or they actually change as genes are turned up and down, are switched on and off, it actually has been a very important question that I feel that it still hasn't been resolved yet. And there's plenty of evidence that pre-looped architecture exists, that a lot of such interactions may in fact be dynamic and may change in terms of how how promoters and enhancers talk to each other during cell differentiation. But there's also some evidence that perhaps physical proximity may not be the deciding factor that says whether an enhancer will switch a gene on or off. And just to go a little bit more on this, for example, what we have learned, as I mentioned, that CTCF and cohesin seem to be very important for the TAT layer of genome architecture. But surprisingly, when you abolish their expression, it seems that there's little effect, or I mean, there is some effect, but there's less than what I was expecting on gene expression. And this raises some questions: how exactly regulatory loops are formed? So also from my work with Giacomo as a postdoc, we noted that here the interactions between enhancers and promoters do not seem to be, at least in the majority, anchored on CTCF, because we don't really see a big difference in the change of interaction strength in context in pairs that do not have a CTCF binding site. So one hypothesis that has been brought forward is that perhaps the specificity that comes with establishing specific cell type interactions is mediated by transcription factors. And there's been some evidence for that in the literature, starting from about 8 years ago with GATA1 and LDB1. And we have also suggested that some transcription factors may play a role in this. But some of the questions that remain is, how exactly is this happening? What is the cause and what is the consequence? And is this really a phenomenon that occurs frequently in vivo in different tissues? So this is like what my lab is interested in. So what we are trying to understand is basically how the different aspects in the different layers of regulation, such as the 3D genome and the chromatin landscape, talk to mediate and inform transcription in order to direct a specific cell fate decision. So we're interested in mechanisms of gene regulation and lineage specification, but we're also trying to understand how this complex interplay can lead to changes in cortical evolution and disorders. So today, what I'm going to talk about is one story in particular that focuses on how differentiation occurs in the cortex in mice. And this is just one aspect of cortical development that I wanted to focus on today. And the way that this works in the cortex is we have neural stem cells, basically. These are these cells depicted here in red that continuously proliferate, but may also asymmetrically divide to generate either another type of progenitors called intermediate progenitors here, or they can also directly generate projection neurons. In most of the cases, the intermediate progenitors will divide one more time and will give rise to two neurons at the same time. And then there's a very specific spatial distribution of these cells, as you can see here on this cortical, on these slices from the cortex marked with the different type, with the different markers of the different cell types. So this is what I'm going to focus mostly today. So how these decisions are made in the cell and what these decisions have at the epigenetic level. So this is a work primarily done by a postdoc in the lab called Boyan, and what we decided to do is, because of the existing heterogeneity in the cortex, where you have many different cell types existing at the same time at different stages of differentiation, we decided to employ the power of single-cell technologies in order to try to dissect how this lineage specification works. So we took one stage in the mouse, so E14.5, and we did it in biological duplicates. We dissected the somatosensory cortex, specifically from embryos, and then we used the 10x genomics to simultaneously profile both the RNA and the ATAC, not from the same cell, but from the same brain. So from the same biological material, we took half of it, we did the single-cell RNA-seq, and the other half we did the single-cell ATAC. And I'm sure that in this network, if you've heard many talks about single-cell RNA-seq, I just wanted to say that it allows us really to quantify gene expression at the single-cell level. To quantify the accessibility of chromatin for different transcription factors in different cells, what we use the single-cell ATAC that allows you basically to identify open regions in the genome that can be bound by different transcription factors. So this is a 3D representation of the data that we actually see at the transcription level. And each sphere that you see is basically a different cell. And what we can identify is known clusters in the cell. So this is, for example, neural stem cells. You can see that these cells represent intermediate progenitor cells. And those are projection neurons in different stages of differentiation. And this is something that I showed you at the beginning. We know that these cell types exist during cortical development, and we can verify indeed with our approach that we can identify most of the types in the cell. So this is very similar to what the classical 2D human projection looks like. So you can compare the two of them, and then you can see that they look indeed very similar. With the 2D UMAP, we can also identify or highlight that there's also other types of neurons that we know it exists in the cortex, such as the interneurons. And we have a very rich data set with about more than 4,000 genes per cell, allowing us to really look at transcription factor dynamics. Okay, so the next question was how this differentiation looks like in our single-cell data. And so this is a single-cell velocity analysis that basically looks at splicing ratios in order to infer what is the future projected trajectory of each cell. So what we can see is that basically neural stem cells are going into intermediate progenitors and then differentiating sequentially to these different stages of neurons. And this is exactly as I mentioned at the beginning. We know that this trajectory exists in the brain. And now, using this kind of analysis, what we are able to do is we can actually ask at what time each of these events in which each gene is upregulated. So here I'm giving you an example. So this is a neurogenin-2, and you can see that neurogenin-2 is expressed in a very specific population of cells. So it starts at certain neural stem cells, and it's upregulated mostly in intermediate progenitors. And using this kind of analysis, combining the trajectory analysis with expression in single cells, what we can do is we can plot the expression of neurogenic in every single cell based on their pseudotime. So where on this trajectory they lie. And this is how it looks like for neurogenic. So what you can see is that indeed there is a very specific time window where this particular transcription factor is upregulated. And this is going to be important for later. That's why I'm spending so much time on that one gene. So if we do this now genome-wide and not just on one gene, we can actually ask what is the transcription, what is the dependence of the dynamics of different transcription factors and different cells. So again, we have on the here on this plot on the x-axis, we have the pseudotime, and on the y-axis, every line basically represents a gene. And what we're looking at is the expression for in each individual cell. So what you can see is that there is a cluster, for example, of genes that are specifically expressed in neural stem cells. So this will be these over here. And then afterwards, there is a sequential waves of transcription factors that are upregulated, then downregulated, and upregulated, and downregulated. And then at the end, in determinately differentiated neurons, we also have a set of markers. So we could use this to infer not only known genes that are important for this process, but also novel genes that may play an influence in differentiation kinetics. So these are two examples. So FHA1 is one of these genes that we predict will be present mostly in neural stem cells, and indeed we verify that using RNA fish, that it's indeed mostly present specifically in neural stem cells. And we could also show that another gene called LRFN5 is present exclusively in mature neurons. And this is a gene that actually has been speculated to play an important role in autism and specifically in intellectual disorders. So next, what we did is we moved on to our single cell ATAC data, and we asked, can we use chromatin accessibility in order to distinguish, again, different cell types that exist in the brain? And the answer is yes. Again, so I'm showing you this 3D UMAP projection, where now we're looking at how the cells differ based on their accessibility profile genome-wide. And again, we can identify similar clusters such as neural stem cells, intermediate progenitor cells, and different flavors of projection neurons. So basically, very, very similar cell types can be inferred from the single cell ATAC compared to the single cell RNA-seq. And the data set that we've generated is one of the highest in terms of the depth, the resolution of the ATAC, which really allows us to look at distal regulatory region. And again, this is how it looks like at the 2D UMAP projection. So one of the questions that we are interested going into this work is like, how much does accessibility differ between different cells within the same cell type? So here's here's how we try to address this. So basically, what you can see here, this is a genome plot. Basically, this is the gene that we're going to be focusing on, DLL1. And here is how it looks like on the promoter of delta 1. So what you can see, these are individual cells here on this part of the graph, and this is an aggregated accessibility this is how it would look like if we were to isolate each of these cell types individually. And what you can see is that the majority of these cells, if you have if this is an accessible region, the majority of the cells show that. What is also interesting is that for example, you can see here, this is now a distal or cis regulatory region, and you can see that this region is also quite specific because it's open specifically in some cell types, but not in others. But when it's open, it appears that most of the cells from the same cell type have an open accessible region. So then we ask the question, if we look at it now genome-wide, for example, can we identify differentially accessible regions and what are their characteristics? So this is what you can see here on this plot. So what we're looking at is basically the average accessibility per cluster. So we aggregate all the cells that are contained in this cluster. And we're asking like what is the differences in accessibility? So you can see that, for example, here, this will be the accessible regions that are accessible in your stem cells and so on and so forth. And surprisingly, what we find is that the majority of differentially accessible regions are actually enhancers. and not so many promoters. There are some promoters, of course, but the majority of the distal regions that change accessibility seem to be enhancers. We've quantified this, and we can see that this differences in these statistically significant, suggesting that again the majority of the rewiring that occurs at the chromatin level occurs at cis-regulatory elements. And here's one example that I think illustrates this point very well. So I showed you the gene FHL1, right? And this is expressed, as I said, predominantly in neural stem cells. And what you can see here is again that the promoter of FHL1 doesn't change at all. It's as accessible in these neural stem cells as it is in neurons where the gene is not expressed at all. What changes are the distal regulatory regions that potentially control, or the distal enhancers that would potentially control the expression of FHL1. And again, this suggests that the variance that occurs is primarily encoded in distal regulatory elements. So the reason why we wanted to do this is because what we want to ask is, okay, if the promoter accessibility doesn't change, how can we actually assign which enhancers potentially regulate the expression of which genes? And the way that we decided to do this is by integrating the single-cell RNA and the single-cell ATAC data, as I mentioned before. So what we do is essentially we computationally align the two spaces, so the single-cell RNA and the single-cell ATAC. And what we can then ask is how the accessibility of each regulatory element, each distal regulatory element, changes in each cell compared to the expression now, not the accessibility, but the expression of the any potential target gene. And this is how it looks like. So I'm showing you basically is that the pairs of either enhancer on the left or gene on the right, gene expression on the right, where the change in accessibility correlates with the change in gene expression. And again, here's what you can see. So these are, for example, the what I'm showing you on the left is the accessibility per cluster at each of these enhancers. And again, we can infer specifically, for example, these will be the neural stem cell specific enhancers. And based on this analysis, we can now say that we predict that these enhancers will regulate these genes because you see that the change in gene expression of the matched pair is very, very similar. So the change in accessibility is highly correlated with the change in gene expression. This is expected. We want to find that, right? So we want to identify pairs where these two measurements are correlated. So why are we doing this? What can we learn from this? Well, what we can learn is we can use it to identify potentially novel enhancers that regulate gene expression. So again, I'm showing you here this genomic plot. This is the promoter of RND2, which is a gene that is expressed specifically upon differentiation. So it's in these intermediate clusters, intermediate progenitors, projection neurons 1 and 2, and then it's downregulated. And what we can identify, so these loops represent now our predicted enhancers that are correlated with the expression of RND2. And we're very pleased to find that some of the known enhancers of RND2, like this one, for example, we can identify very clearly that indeed is accessible only in cells where RND2 is expressed. But we can use this approach in order to identify enhancers that were not known for RND2. Like, for example, these ones, for example, they were way too far to be able to be predicted with the conventional nearest gene approach. But we see very, very similar dynamics in terms of their accessibility compared to the known enhancer for RND2. So what we can then do is we can ask, for example, okay, what are the potential mechanisms? So what are the transcription factors that may be associated with this change of accessibility at different enhancers? So here what we're looking at is the motif enrichment analysis for each of these individual types of enhancers. So for example, for neural stem cells, what you see is that transcription factors such as CAT2 and FOS-Jun that are enriched in these neural stem cell enhancers, and other types of factors such as neurogenin-2 that are actually depleted from these kind of enhancers. Upon differentiation, we now observe that some of them switch. You can see that now neurogenin-2 appears to be much more enriched in IPC enhancers, while others are again downregulated. So there's quite a lot of dynamics that are associated with the accessibility of transcription factor motifs during differentiation, and this can tell us something about what are the potential drivers or molecular mechanisms that can mediate such a change in accessibility. So the next question that we asked is a little bit more complex. So I'm going to spend a bit more time here to try to explain to you what we are asking. So the question that we asked is, does the accessibility of the linked enhancer comes before or after the change of gene expression of its matched gene? So if we look at this graph, so the first part, if you focus on this part over here, what we're plotting is basically the accessibility of each enhancer. So each row represents an enhancer. Each column represents a cell that is ordered based on our trajectory analysis. And what we can say is we can indeed order the enhancers based on their maximum accessibility. And what you can see is that, again, they're very specific. So you see that this cluster of enhancers, for example, again, is neurostem cells. This one represents a change in accessibility during differentiation and so on and so forth. But now what we can do is we can ask, what is the difference between the maximum accessibility of that particular enhancer and the transcription that occurs in the linked gene? And this is what we're looking at in this part of the graph, where we're basically asking if we subtract the two values, so the maximum accessibility of the enhancer and the maximum time when the expression occurs, we can infer a value which we call the delta pseudotime. And if this value is negative, it means that the enhancer accessibility occurred before the maximum of gene expression. And what we find is, interestingly, for cells that are undergoing differentiation, so for transitional stages, we see that this is indeed true genome-wide, so that the overall you see enhancers where the accessibility of the enhancer coming before the change in gene expression occurs. Interestingly, in cells where they have already been defined, such as neurostem cells or neurons, we don't see the same phenomenon. So this seems to happen primarily in mostly in differentiation conditions. Okay, so here's one example for how it looks like at one gene called Ailmes. And here what you can see is basically these are all the individual enhancers of Ailmes, and we can see that there's the majority of them here. This is the aggregate, and the accessibility of the enhancer comes first. The expression of the gene comes a bit later, but true priming actually seems to happen only rarely. So only some enhancers actually have this very specific priming early on. Okay, so in my remaining time, what I'm going to spend is to look at how the changes in gene expression are correlated with changes in 3D genome and DNA methylation. So what we did is we basically used an approach to sort the different major cell types in the brain. We did FACS, and then we did a combined DNA methylation with Hi-C in order to look at both aspects at the same time. So I'm going to skip this briefly, and I'm going to go to this part because I wanted to tell you what we found in terms of correlation between gene expression and 3D genome architecture. So what we're looking at is the two types, so the positively correlated enhancer gene pairs that I mentioned before, and our controls where the change in accessibility does not match the change in expression. And what we're asking is, how is the 3D genome architecture associated with of these two pairs? And what you can see here, so these are aggregate plots where we can quantify genome-wide the interactions between the enhancer and the matched gene. And you can see that for neurostem cells, particular pairs, you see that the interactions are the strongest in neurostem cells, and then they decrease in intermediate progenitors and projection neurons. This is not the case for control pairs, suggesting that indeed we see a very specific phenomenon associated with 3D genome architecture in these cells. We quantified this, and we can actually see that this effect is very specific. So you see that overall, the context between the enhancers and promoters are specific for cell type. This is not the case in control pairs. However, are they actually really all the same? And this is a major question that we have in the field. So here what I'm showing you is each dot represents a single pair of enhancer and promoter, and they're either, the interaction strength is shown here on the X and the Y axis in these two cell types. So what we show here is that the majority of enhancers and enhancer gene pairs, indeed, they seem to follow this pattern. So here's, for example, one example. This means that these two regions contact each other strongly in your stem cells, and then they go further apart in neurons. But this is not always the case. So for example, we see definitely evidence of pre-looped, strongly pre-looped enhancers where the interactions remain even after the gene has been switched on. And we also see evidence for enhancers where there is no, or there's very weak interactions despite the fact that these genes are specifically upregulated. And this is different, for example, with the control enhancers, where we don't see this kind of bias, and it's mostly random. So I'm going to go only briefly for the DNA methylation, where we basically see that the DNA methylation levels of enhancers are, again, very specific. And in neurostem cells enhancers, for example, the DNA methylation levels are the lowest. And as the enhancers become silent, then you can see that the DNA methylation levels increases. This is not exactly the case in controls. I'm going to skip this. And then I'm going to go to one of my last data slides. So how exactly does this work? So what is the potential molecular mechanism that we think drives the changes in genome organization? So the way to address this is what we did is we looked at the correlation between pairs of motifs, pairs of binding sites for specific transcription factors. Here's on the x-axis is the context trend, and on the y-axis is the variability between these interactions. So what you can see is that in this particular, for example, in the upper right corner, transcription factor binding sites that are predicted to have strong context and yet very cell type specific. And I can show you two examples for this. So for example, for Reorganization that we identified with this analysis, we can see that basically the interactions seem to be the strongest in intermediate progenitors. And again, this is the cells where Reorganization is expressed, as I showed you earlier. For PAC6, which is a transcription factor expressed only in neural stem cells, you can see that this is the opposite. So that the interactions are strongest in neural stem cells and then they decrease, but the relative proportion of the maximum is different. So we can do the same kind of analysis for methylation. And what I want to show you is that we also find that Reorganization is associated with low levels of DNA methylation that are also cell type specific. And here's what we can show. So this is a plot that looks at the DNA methylation levels at Reorganization sites, and you can see that there is a difference between the different cell types. Also, the accessibility of Reorganization binding sites is different. So why am I spending so much time on Reorganization? Well, one of the questions, and this is the last data slide, is, I'm going to skip this actually. One of the questions is whether or not we can, the transcription factor is driving the changes in 3D genome architecture and transcription, or is it exploring an already pre-existing architecture? And in order to address this, basically, what we decided to do is to now did in vivo experiment where we electroporate in utero Reorganization in the brains of the embryos, and then 24 hours later we fact sort the cells and we ask what are the changes in gene expression, what is the change in connectivity. So we achieve about 6-fold overexpression of Reorganization, and importantly, we can confirm that we don't have major changes in cell types, so that we don't really have indirect effects. Although we do have a change in one of the predicted targets of Reorganization, such as the illness. So we know that this is working. And then we ask, what happens now with 3D genome architecture? And the answer is that Reorganization overexpression appears to be sufficient even within 24 hours to upregulate the interactions that are centered around Reorganization, meaning that it's not just a consequence of the rewiring of the 3D genome, it actually has a causative effect. We can quantify this and we can see that indeed this effect is highly significant, although it's not in absolute terms, it's not super strong. It doesn't mean that all the time the two regions will be in contact. But this is very similar to what we would find in the endogenous situation. So the difference between the control condition and the Reorganization overexpression condition is very similar to the difference between what you would actually find between the two cell types in vivo, suggesting that at least we think that the majority of the changes of the 3D genome at enhancers seems to be mediated by Reorganization. Okay, so to summarize what I've told you today is that I've showed you some evidence that integrating different types of data, specifically single cell epigenomics with cell type specific 3D genome or DNA methylation data, can tell us something new about how different cell types are generated and how cell fate decisions are made. And the important point here to remember is that we can't really infer neither the 3D genome information nor the DNA methylation purely from single cell ATAC alone, because we see this difference in terms of the absolute values of connectivity. The other, one of the other conclusions is that it seems that tissue specificity at the chromatin level is encoded mostly in distal regions. And it seems that the regulatory context in DNA methylation levels at enhancers are mostly cell type specific, meaning that they engage in specific interactions, but this is not the only rule that exists. So we see evidence also for prelooped enhancers and promoters. We also see evidence for pairs that do not seem to engage in physical context, suggesting that actually it's probably more complicated that we thought at the beginning, and then maybe all three models are correct, at least to some extent. Nevertheless, importantly, overall, there is the majority of them are dynamic. And then finally, I show you that the temporal waves of transcription factors seem to drive the reorganization of the epigenetic landscape in 3D. And I gave you evidence for neurogenin 2 specifically that it seems to be doing this and also actually mediating 3D genome interactions. But we also think that other transcription factors may be present in this. So this work has been posted in the bioRxiv very recently, so a couple of months ago. If you're interested, I encourage you to go there and to look more. And this is my lab. So the most of the work that I showed you today is done by Florian Noack, the postdoc in the lab, with the help of Sylvia, Jay, and Faye. And yeah, and then with that, I want to thank specifically the Pioneer Campus for giving me the opportunity to establish my lab and for the generous support, also the other funders, and you for listening. Thank you for the attention.
Thank you very much, Boyan. I'm sure there will be a lot of clapping if we would be in a normal place now. Actually, there's quite a lot of questions in the chat, so I will start in order with them. Maria had a question, and I think I will unmute you, Maria, and you should be able to talk if it works. Yep.
Am I muted? No, that's it. Okay.
Yeah.
Thank you, Boyan. It was a beautiful talk and beautiful, amazing data that you have. I guess I have the obvious question, which is when you merge or you align the ATAC and the RNA-seq data, how do you actually do it? And how do you make sure that whenever you match ATAC and RNA, you know, whether you use openness in promoters or openness in cis-regulatory regions or gene activity or whatever you use, but then when you compare the ATAC and the RNA and you see that, for example, the openness at enhancers happens before or after the transcription, that this is not an artifact of the way that you design the data?
Yes, absolutely. This is a very good question. So the way that we do the aligning of the two spaces, so this is based on a method first developed by the Greenleaf Lab. So Granja developed it, I think it was about last year or a year and a half ago. And what this method does is basically it once the two data sets are integrated, and I'm going to talk here a little bit about your second question, which is how do we decide on what to align? We actually tested this. So we looked at, for example, what is the best way to integrate this? So if we use, for example, accessibility that is specifically on promoters, if we use accessibility that is based on the whole gene body, or if we even incorporate things like Cicero or things like, for example, matched gene activity, what is the best case scenario? Because there's been many suggestions of how to do this best. In our hands, it seems like looking at the accessibility across the whole gene body is the best way to do it, and it's the thing that corresponds the most to the real gene expression. Because as I mentioned, looking specifically at promoters don't seem to change that much, at least in our systems, or perhaps in other systems where the cells are terminally differentiated, promoter accessibility changes, but in our hands, it's not the case so much. So we don't really gain so much information if we focus only on promoters. If we incorporate the enhancers, then of course you're biasing it a little bit towards the enhancers. So that makes it difficult to say later on, as exactly as you said, how to match it with the enhancer accessibility. So we did it over the gene bodies, and we tested all the other possibilities. These seem to give us the best result in terms of the integration compatibility. Now to your second question, how can we be sure that the patterns that we see are actually not an artifact of the correlation? Well, one possibility is, of course, is to look at each individual enhancers, as I showed you, and we can see that in our data, we can definitely see enhancers that, although correlated overall, we see that the enhancer, for example, the omics enhancer that I mentioned at the beginning, is accessible before actually the gene becomes expressed. So it doesn't mean that we require an absolute correlation all the time. It's just we just require a threshold. And the second answer to your question is that in recent work by the Boyan lab, they actually done it in the same cell and without any computational integration. And you see very, very similar dynamics. Indeed, that the majority of the enhancers seem to be coming open before gene expression. Now, one caveat here, and I'm going to stop because I'm sure there will be more questions. One caveat here is that we're talking about mature mRNA. So we're not talking about nascent expression. So it could be that if you look for some of those genes at nascent transcription, it could be that this comes slightly earlier, and then we don't really know how much exactly is the lag between the accessibility and the nascent RNA in terms of transcription. So I think that this will be a very important question to address going forward.
Yeah,
okay.
Thank you, Boyan. There's quite a few questions indeed, actually. So Ines, I'm going to unmute you. So if you can now ask your question. Ines? Yeah. You should be unmuted already, Ines.
I don't hear anything.
No, me neither. Okay, so I don't know if you can hear us, Ines. If you no, okay. So maybe what I'm going to do is I will go to the next one, and then maybe Ines, can you write it on the chat, and then I can read it afterwards if that's okay for you. So we have also a question by Robert Simpson, and I'm also going to unmute you. There you go.
Hey, this is Rob. How you doing?
Hi.
Wonderful talk, Boyan. Thank you so much. It was great. I have a small technical question for a clarification, then an actual question. For the technical part first, the enhancer priming, what kind of time scale are we talking about here? And then for the question, when you look at the correlation between accessibility and transcription, do you actually see any, or have you looked for any specific sets of genes that do anti-correlation in that respect, where you would see something like direct repression? And what does that mean for the promoter if you see that?
Yes, these are both excellent questions. I'm going to start with the second one. We can definitely do, and we have done this same kind of analysis, but now looking at anti-correlated pairs, so where the accessibility of the distal element is actually anti-correlated with expression. We haven't explored it as much, and I think that this is something we should definitely look into. We see that there are different sets of transcription factors that are where the motifs are enriched in these, so they're not the same as what we see on the positively correlated. There are some repressors, but I'm a bit hesitant to say really what is the cause and what is the consequence there. So we're not entirely sure how to look at this. One thing that I can say is that we see that, for example, the anti-correlated pairs, the distance between it's not an enhancer, but let's say the regulatory element and the promoter seem to be much smaller than the positively correlated one. So for some reason, these regions where the enhancer may have a negative effect on gene expression, they appear to be much closer to the actual promoter than the normal positively correlated enhancers. Now, for your first question, so what is the time scale in terms of this priming? Difficult to say, because what we can, I can tell you the pseudo time units, but how much does this exactly correspond to real time? We don't really know. It seems that for some, it can be in the scale of hours. It can be also slightly earlier. For majority of them, it's just perhaps like maybe an hour or two. But again, this is mostly speculation. What we do see is that all of this happens within one cell division. So there's not a huge lag in terms of 10 or 12 hours or so on. So this is really a relatively quick, but how quick, I can't really say.
Thank you so much.
So the next question I will read is by Simon Rosowski. Which do you use for the integration of single-cell RNA and single-cell data?
I didn't hear the first part. Which tool? Or Maria, I didn't hear. Can you hear me?
Yeah, I think my internet is going a bit, so I'm going to repeat. The question is on the by Simon Rusovsky. No, you can't hear me. Okay.
Now I can hear you. Yes.
Okay. So the question is, which tool have you used for the integration of single-cell RNA and single-cell ATAC-seq data?
Yes. So as I said, the tool that we used is developed by the Greenleaf Lab. At the time when we did the analysis, this was based, so the tool wasn't yet available. So we did it based on the description of in their paper. So this is the Nature Methods paper from 2019 by Granja et al. Now they have also, the Greenleaf Lab has released a tool that can actually, you can use yourself to merge the two data sets, and the tool is called Archer. It's very, very similar to what we did. The differences are like just that we used a slightly larger window because we wanted to actually quantify enhancers that may lie further than their default, which was the 250. So we looked at the window of plus minus 500 kb, meaning that we may underestimate also some enhancers that are further, even further than 500 kb.
Good, thank you. So the next question is Pia. Pia, I have unmuted you, Pia Rautenstrauch. The first question was already answered, but if you can ask your second question, I think that would be great. No? Okay, I will read it then. And then I think probably she can listen to it. So it seemed that you had a more fine-grained annotation for your single-cell RNA-seq data. Were you able to recover some of this granularity in your single-cell ATAC-seq data using single-cell RNA-seq through your integrative analysis?
That's a very interesting question. Indeed, it seems like some clusters, like for example, for the neurons, we see 3 clusters at the RNA level compared to just 2 clusters at the ATAC level. And some of the differences are actually not as significant as they may appear, because one of the major differences between the RNA, what we see at the single-cell RNA and the single-cell ATAC, is that the single-cell RNA distinguishes between cells that are currently undergoing mitosis, so G2M mostly, while at the ATAC level, you don't see actually a difference between cells in the different phases of the cell cycle. And this to me was surprising at the beginning. I would have expected that this is not the case, but there is plenty of evidence in the literature using fact-sorted populations from different phases of the cell cycle and doing ATAC, is that indeed you don't see major changes in chromatin accessibility during the cell cycle. So that's one of the reasons why the two things appear different. When we looked at different data sets, so much bigger data sets than the one that I've currently shown you, we see that sometimes in ATAC, you can see differences that you don't really see in the RNA. So it's not a one-way street. It's not that whatever you see at the RNA gives you higher resolution. No, sometimes you see differences that appear in cell types which appear homogeneous at the RNA level, but at the ATAC, you see they seem more different. So I don't think we should really try to make a rule for this.
Okay, good. Thank you, Boyan. So next question is by Jeremy Breda. Concerning the time delay between accessibility and expression, wouldn't the cross-correlation be a more appropriate measure?
The cross-correlation, yes, I mean, it can be, of course, and the caveat there is definitely that we're looking again at so something that Maria alluded at the beginning, that we're looking at at enhancers that we ask them to be correlated with gene expression. So there may be a subset of enhancer gene pairs where we're missing this, or where if this is really truly off, if the enhancer, for example, is accessible, I don't know, 5 or 6 hours before the gene, and then it switches off and it's totally uncorrelated with gene expression, we would miss this, and this is a caveat of this kind of experiment. But it's very difficult to envision another way to correlate it. So we would have to impose other assumptions, like for example, that the enhancer is in physical proximity of the gene, which again, as I showed you, it doesn't necessarily mean that this is the case. So I think that this is a very open area in terms of what is the best way to do it. We think that with this kind of correlation, we can recover the majority of the enhancer pairs, and they seem to be important, but at the end of the day, what matters is whether they're functional or not, and this is something that we're exploring at the moment.
Thank you. I think there was another question by Ivan, but I think he's not raising hands anymore, so I'll go to the next one. So the next one is by, this is long. The question is a little bit long. So this is by Abdul Rahman Salhat. The question is, thank you for the very interesting talk. You have explored DNA methylation at distal regulatory regions and tried to link it to the 3D genome architecture. My question is, did you have a look for genome-wide DNA methylation on a larger level like TATs and or AB components?
Yes, this is probably something that I did miss because I skipped over it, but I won't go back to the slides. I will just tell you. Yes, we definitely looked at it. One of the reasons why we looked at that is because we see an increased installation at the TAT boundaries upon differentiation. So we see that the TAT boundaries don't change much. There is some changes, but similar to what I've reported previously, about 10% or 50% of the boundary changes. But what we see is that the ones that remain, there's an increase of installation in the neighboring TATs. So one of the possibilities was that this is associated with lower levels of DNA methylation, maybe that is, or higher levels of DNA methylation. So we explored this basically whether there's any change at DNA methylation levels at the TAT level, and the answer doesn't seem to be that is the case. We don't see global changes in DNA methylation in these structures. In compartments, that's a little bit more tricky to say. So we looked at that, but compartments usually aggregate regions that are very big, so like hundreds of kbs or so. And we see some changes in there that are typically correlated with changes of gene expression. But again, I'm a bit hesitant to say that this is indeed a true correlation and what is the cause and what is the effect, because it could be that because you change gene expression, that is maybe why you see these big changes in the compartment, and this is why you see changes in DNA methylation levels. But overall, at the TAT level and the TAT boundaries, we don't see changes. When you look at CTCF sites, for example, which is one of the things that was interesting for us, we don't see changes in the DNA methylation at CTCF sites. So that's I think means that the majority of the changes of DNA methylation occur at distal elements, because also at promoters, we don't really see changes in DNA methylation that are very strong. So most of the promoters are hypomethylated, and they remain hypomethylated independent of what happens with gene expression.
Thank you, Boyan. And I think I'll take just one more question. This is from Ignacio Ibarra del Rio, and I have already unmuted you, Ignacio. So if you want to ask your question, please go ahead.
Hello, morning. I think this question was already answered, but maybe I can phrase it differently. I was wondering, the enhancer promoter contacts you have and associating those with HiC scores, what is the recall of known HiC contacts that you have when overlapping those two? Are there too many like highly correlating peak pairs that do not have HiC? And is this something that is is enriched for HiC, but overall you have many regions that are highly correlated but without HiC loops?
I'm not sure I understood your question exactly. I mean, the HiC score that we use, this is a measurement of the contact frequency. So it's a measurement that takes into account both the coverage and the distance between them and normalizes it using the method first developed by Amos Tanai. So it's not a pure measurement that is based on either just the distance between the elements or the coverage, which are the two major biases in HiC data. So when you see HiC score, you can think of it basically as an estimate for contact enrichment or for contact frequency, right? And again, another thing that, I mean, if I understood your question correctly, how does this compare to the known contacts? So first of all, what is a known contact, right? So the ones that we have previously shown, for example, with the very high resolution HiC data in the cortex, we see that a lot of them are very similar. And so, for example, you clearly identify CTCF loops, you clearly identify any kinds of normal, what we would consider, let's say, a stronger and more architectural types of loops. So these are very clear, but they are not what the enhancer promoter contacts are like. So the enhancer promoter contacts are relatively weaker compared to the conventional CTCF convergent loops. And that's why I think if you don't have enough resolution and if you don't properly account for the biases in high-C data, sometimes you may not see them. So this is, I think, very important point to make here, that the correct type of analysis is very important if you want to really link expression to 3D genome connectivity. I'm not sure if this answered your question, but
it does. Thank you.
Very good. So I think I'm going to finish here because we are almost at the hour, and just to encourage the couple of questions that are left by Menghan Wang, maybe you can directly write to Boyan. I'm sure that he'll be very happy to answer. And with that, I really want to thank you, Boyan, for sharing your data and the very exciting new data sets that you have, and to all of our participants for staying here and for asking all the questions. And as said, in 4 weeks' time, Wolfreich, December 4th, please stay. And again, thank you, Boyan, for the talk, and thanks to everybody. Have a good day.
Thank you, everyone. Thank you, Mariana. Bye.